Genetics Matters, Episode 12. A single strand of DNA is a powerful tool. Hello again, I am Brad Goodner. When we were last together, we learned how scientists have used DNA replication in a variety of ways as lab tools. The biggest of these replication-based applications is PCR, or polymerase chain reaction, which allows one to make millions of copies of a specific sequence by initiating DNA replication on either side of that specific sequence. All we really need to know is the sequence of two primers, single-stranded DNAs of nucleotide residue lengths in the low to mid-20s. Each of these primers can bind through complementarity to their target site when the target DNA is denatured. That is to say, when the target double-stranded DNA is forced to become separate single strands. PCR primers are one example of the power of a single strand of DNA, what I like to call a probe. The word probe makes us think of investigating, of finding new things, and that is exactly what a DNA probe can do for us. The simple act of a probe interacting with a complementary sequence through base pairing allows us to find those complementary sequences in a large mixture of mostly unwanted sequences, like a needle in a haystack. All we need is a way to make our probe visible. In the ancient days, when I was a graduate student, in the 1980s, we labeled our DNA probes using radioactivity. What I mean by that is we used radioactive phosphorus incorporated into the 5' phosphate group of a nucleotide. Radioactive labeling is still used today, but much less so than 30 years ago. In the 1990s, two additional ways to label DNA probes became available. First, certain enzymes with easy-to-follow colorimetric assays were overproduced through recombinant DNA technology, and methods were established for how to tag enzyme molecules onto a single-stranded DNA probe. Once the DNA probe had bound to its complementary target DNA strand, the location of the DNA probe was determined by adding the appropriate substrates acted upon by the probe-linked enzyme molecules. Second, small fluorescent molecules were discovered or created that would absorb light energy of one distinct wavelength and re-emit via fluorescence light energy of a distinct longer wavelength. These fluorescent tags can be attached to a single-stranded DNA probe and found by excitation with the appropriate wavelength of light. Let us see the value of a single-stranded DNA probe in action. Imagine that we have cloned random chunks of an entire genome into a plasmid and introduced these different plasmid clones, we'll call them, into E. coli to replicate and maintain them. Each E. coli cell can potentially contain a different clone chunk of the genome. This is called a genomic library. If the target genome was, say, 1 million base pairs in size, and I cloned 1,000 base pair random chunks to make the genomic library, then I would need a minimum of 1,000 E. coli colonies to cover the entire genome, if, and it's a big if, I could hand pick which clones I needed. I don't want to have to go through all the different library clones to find the right 1,000 that together would cover the entire genome. So it would be easier if I could keep, say, 5,000 randomly chosen clones, because based on a statistical distribution called the Poisson distribution that deals with random events, 5 times the minimum number to cover the entire genome will give me a 99% confidence that every part of the genome is covered at least once, somewhere in those 5,000 randomly chosen clones. Sort of like pouring out from a bag of M&Ms in which there's 8 colors. I know I'm not going to get all 8 colors by just pouring out the first 8 M&Ms. I have to pour out a few more. Actually, I should pour out about 40 to be 99% confident I have at least one of each color. Certainly I'll get lots of blues and maybe more browns than I expected, but I'll at least get one red. That's the idea. So I picked the 5,000 E. coli colonies and patched them in an organized array on fresh agar growth media in petri dishes, say 50 colonies per dish, for a total of 100 petri dishes. I let them grow up overnight. Now I take circles of a special paper that are just smaller than my petri dishes and can bind DNA, and for each petri dish I carefully lay the paper circle straight down on the agar surface to pick up some of each gridded colony on the plate. I pick the paper circle straight up and lay it colony side up on the surface of a solution that contains detergents and enzymes that can burst open the E. coli cells. After enough time to break open the cells, I slide the paper circles over to a new solution at a high pH that will cause double-stranded DNA to denature, to become separate single strands. Both the E. coli chromosomal DNA and the genomic library plasmid clone DNA will denature in each colony spot. I then dry the paper circles, and I can either bake them or expose them to a little bit of UV light to get the DNA single strands in each colony spot to bind to the paper. Once the DNA is bound, I can gently wash the paper circles to get rid of most of the dead cell debris. Proteins, carbohydrates, lipids. I don't need those. I have generated what are now called colony blots, and I can probe them. Let us say I want to find the genomic library clone or clones containing a particular gene. I have a labeled single-stranded DNA probe. I add the labeled DNA probe to a solution containing my 100 colony blots. I control the temperature so that the labeled DNA probe can bind to its correct complementary sequence whenever it randomly bumps into it, but it will not bind to sequences that are not perfect or near perfect complements. After allowing sufficient time for my labeled DNA probe to bind to complementary sequences on the colony blots, I gently wash the blots to remove any probe that is non-specifically interacting with the blots. I can control the stringency of my washes with temperature and/or salt concentration. The higher the temperature of the wash solution, the more perfect the complementarity has to be between the probe and its target. Now, too high and even of a temperature and even 100% complementarity will not be strong enough to hold the probe to its target. In terms of salt concentration, because positively charged ions, cations, in the solution can charge shield the negative charges on each DNA strand, the lower the salt concentration in the wash solution, the more perfect the complementarity has to be between the probe and its target. When I'm done, I look for the labeled probe, and hopefully one or more colony spots will light up on one or more of the paper circle blots. I go back to the corresponding petri dishes, because I was smart enough to label the petri dishes and their corresponding colony blot papers, and I pick the proper E. coli colonies to grow up and isolate plasmid clone DNA containing the gene of interest, all because of a single-stranded DNA probe. In that example, our probe was free to float around while its potential targets were bound to a surface. But we can also flip it around. Suppose I want to make a genomic library from organism X, but I only want sequences that are not also found in organism Y. I take genomic DNA from organism Y, break it into roughly, say, 1,000 base pair chunks, make it single-stranded with heat or high pH, and then bind it at one end to a surface, say some tiny beads. Now I take genomic DNA from organism X, break it into roughly 1,000 base pair pieces, make it single-stranded, and then I incubate it with the beads dangling with single strands from organism Y. Base pairing occurs between complementary sequences at a stringency that I can control with temperature and/or salt concentration. And then I spin down the beads, taking away not only the organism Y strands, but any organism X strands that are complementary to those organism Y strands. I move the liquid away from the beads, and that liquid contains unbound organism X sequences. Do this a couple more times with fresh organism Y beads to make sure I've found all of the so-called cross-complementary sequences, and I will end up with sequences unique to organism X. I have made what is called a subtractive library. Single-stranded DNA probes can be used in all sorts of ways. We can look for specific pathogens in clinical samples from sick patients. DNA probes can be used to determine if a blood stain is from a human or not. It is amazing what the simple act of complementary base pairing allows us to do in terms of DNA-based technologies. See you next time on Genetics Matters.